Good afternoon, everybody. I'm Stephen Burley. I'm the director of the Institute for Quantitative Biomedicine, and it's my pleasure to welcome you here today to the seminar that's being given by Professor John Chodera from Memorial Sloan Kettering. I've got a few housekeeping announcements before we start. John is the third of the speakers for this series. We'll be hearing from one of our colleagues, Judith Penzis, next week, and then on the 8th of November, I'll speak. And then we'll round out the program with three additional speakers, one of whom is a former tenant of the building from Renew CO2, Anders Larsen, whose biotech company or bioengineering company is going on to Renew CO2 Incorporated is going on to bigger and better things, having raised additional funds and received a very substantial incentive payment from the Gates Foundation. So they were doing something right upstairs. With respect to climate change. Sorry. I remind you that October 31 is the deadline for applications for the IQB Inclusive Leadership Award, which is held this year by Jennifer Jiang from the Wei Dai Lab. For details, please contact Michelle. Emails provided on this slide. This is the week where we wish both Irving Geis, who is the artist who generated the images that are on display in the Geis Gallery, and the Protein Data Bank, happy birthday. So Geis was born on this day in 1908 and passed away in 1997. And I had the pleasure of getting to know him and becoming his friend and even commissioning a painting from him. He was a pioneer in visualization of biological macromolecules and a force in terms of textbook publication, etc. The Protein Data Bank, as you know, was established as the first open access digital data resource in 1971, and that the announcement formally came in Nature New Biology on the 20th of October of that year. So 52 years of continuous operations, 52 years of continuous federal funding for the Protein Data Bank. Not a small feat, reflecting the central importance of the archive in biomedical research. A couple of announcements. Tomorrow at 2 p.m. we'll be collaborating with the American Society for Biochemistry and Molecular Biology, the president of which is in the audience. Anne Stock, thank you for your service, Anne. We will be featuring an opportunity to learn about teaching enzymology with the Protein Data Bank, going from the pandemic to Paxlovid, and more precisely to nirmatrelvir, the active ingredient of Paxlovid. I also want to draw your attention, undergraduates and graduate students, to the 2024 bootcamp that we'll be hosting here in the winter session on science communication. The applications are due via email on the 31st of October, and I urge anyone who's interested in honing your skills in terms of writing, scientific writing for the lay public, and molecular visualization to consider applying for the bootcamp. It is a rewarding experience for everybody who's involved. And more generally, I want to reiterate that there are opportunities for both professional appointees and graduate and undergraduate students here at the RCSB Protein Data Bank, and there's a list on this slide of some of the opportunities that are available on our jobs page, www.rcsb.org/pages/jobs. So with that, I'll finish and introduce our speaker. So it's a pleasure for me to be able to host Professor John Chodera from Memorial Sloan Kettering, where he was recently promoted to the rank of full member, which is the equivalent of full professor here at Rutgers. Our careers have crossed in various ways over the decades. John did his bachelor's in biology at Caltech, and then did a PhD with Ken Dill at the University of California, San Francisco, and it was working with Ken that I was able, with others, to co-found Prospect Genomics, which was acquired by SGX Pharmaceuticals, which set the stage for me going to the dark side to industry for a decade of very productive interactions. After a very brief postdoc at Stanford, with the Folding at Home project with Vijay Pandey, John was appointed to the very prestigious position of Independent Distinguished Postdoctoral Fellow at University of California, Berkeley. This is equivalent to a Harvard University fellow position. He went from there, having been very successful, to an opportunity at Memorial Sloan Kettering Cancer Center in New York City, where he's been ever since, making his way up through the ranks. And as I said, he was just promoted a very short time ago. A well-deserved, and I would say somewhat overdue honor, but it is what it is. John's lab is developing the infrastructure to enable fully autonomous small molecule drug discovery powered by next generation hybrid physical and machine learning tools to predict potency, selectivity, and resistance to small molecule pharmaceutical agents. And I'm looking forward to the talk. Thank you very much for coming and making the effort to visit us here at Rutgers. It's a pleasure.
Thank you so much. It's a pleasure to be here, and it's a great opportunity to tell you about some of the work going on in my lab. I've had a great day visiting so far, especially the opportunity to meet some rock stars working with the PDB. We are big consumers of a lot of the work that they do, so it's really great to meet them in person. I first want to start off with who I am and where I am. So I'm John Chodera. I'm part of the Computational and Systems Biology program, which is what we call a department over at Memorial Sloan Kettering Cancer Center. We've got a number of other fantastic scientists working in this fancy new building, the Zuckerman Research Center, in the Upper East Side of Manhattan. We're right next to Rockefeller University, and a lot of our graduate students come from the Weill Cornell Graduate School of Medical Sciences. It's a phenomenal place. We're hiring faculty every year, so if you're interested in faculty jobs in computational biology, and broadly spoken, we're happy to have you. You don't have to work on cancer. It's just good biological research. So we use a lot of interesting tools in the lab to tackle different problems in building new technologies for drug discovery. A lot of those I'll tell you about later today. Some of them include molecular modeling, like you might see David Case or Darren York doing. Some of them involve integrating machine learning at every aspect of this, from learning flexible functions for describing molecular interactions, all the way to learning surrogates for how small molecules interact with proteins or different tox targets. We use these graphics processors that you probably use to play computer games, and then we have these distributed computing networks that we also work on. I'll tell you more about that a little bit later. But we also have an automated wet lab. This is our robotic undergraduate. It moves plates around back and forth to do different kinds of biology experiments. We mostly generate data that it's too boring for other folks to generate. So we have to invest in doing some of this ourselves with robots. And then we also excited about how to bring that technology to $5,000, almost disposable Opentrons robots that are artisanally crafted in Brooklyn, where I live. And so these things are powered by Python. They can do lots of experiments. They only cost $5,000 a piece, so you can buy as many as you want. Or you could use things like cloud laboratories like Stratios or folks like that are trying to automate and industrialize and become the AWS of these technologies. So my lab works on a wide variety of problems. Mostly, we're working on challenges for which there's no good predictive models, where industry has a problem, and in order to make drug discovery work better, they'd like to come up with more predictive models that use physics and machine learning in some broad way. And so a lot of that has been, because I'm at a cancer institution, working on selective inhibitor design. There's 500 kinases. They all look alike. People target their ATP binding sites. You need to distinguish between them, so you need a selective way of designing small molecules that are selective for the ones you want. They also need to be potent. They have other drug-like properties. And then you have to deal with things like resistance. So after a while of dosing these things, you'll see resistance emerge, lots of different mutations. And if we could predict that with biophysical models, because we're seeing this evolution happen inside of cells, then that would be phenomenal. And that's different things that we're working on to try to ideally design inhibitors that are not going to be subjected to mutational mechanisms of resistance in the lifetime of the patient. And then there's lots of other cool stuff we're working on. Some of it was with awesome collaborators working on nanotechnology for drug delivery. We're really interested in making the most important or useful or informative biophysical experiments happen. And so we're working on how we interpret and automate those experiments. And then a lot of cool stuff is going on with the design of biologics right now. So one of the amazing things about what we get to do is that we get to build open source software, and we get to collaborate very broadly with industry to do this. So they're very happy to work with us, and we've had a huge number of different industry folks who've come to us with problems. We get to collaborate with these scientists. It's great for the graduate students who get exposure to what's going on in industry, and it's super exciting to actually work on problems that are impactful. Drug discovery is really hard, so anything we can do to make it easier is, of course, very beneficial. We also work with all these other larger organizations that help build pre-competitive tools, open source software that helps make drug discovery easier for everyone. And then finally, all of these fantastic data generators that generate new data that will hopefully be useful for helping us make this problem more tractable. So did anyone else have an interesting pandemic? It's certainly been an interesting few years. So what I'm going to do is I'm going to take you in a time machine and take you back a few years to February 2020, when the New York Times said, we were a little bit worried that this coronavirus epidemic that has emerged elsewhere is going to come to the United States. And of course, being the United States, we have to be #1 at everything. So very quickly, we were #1 in number of coronavirus cases anywhere in the world. And this got a lot of folks thinking, probably many of you in the audience as well, what could we do to aid this response, right? We think about biology and disease. Surely there's a way we can put our skills to actually be useful. Some of us who design small molecules have been thinking all along about, we're going to really need oral antivirals. These are antivirals that you can deliver. You can pop a pill, you take them when you get sick. You could possibly even use them prophylactically if you've been exposed. People do this for HIV, for example, and other diseases. One of the worries we had is that if we didn't know how long it would take vaccines to actually emerge. And one of the amazing things was it was much less time than we thought using these new mRNA platforms. But we'd also need complete safety if vaccinating 100% of the people. And it would also be very difficult, especially in this country, to get to 100% vaccination rates, the kind we need to really be protective. That's a huge problem, much more than we anticipated. One of the other problems is that vaccines target, of course, these highly variable exterior regions of the virus, and that you have to keep up with the viral antigen drift and escape, which is proving to be a huge challenge right now. So if you had an antiviral that could target things that were much more stable, much more conserved, maybe you could avoid that kind of thing. And then, of course, the first vaccines from Pfizer had -80°C cold chain storage requirements, which is very difficult to make it globally accessible. So one of the cool things was that data has moved so quickly during this pandemic and has been shared much, much more rapidly than publications. We didn't have time to wait for publications because people were dying. And I think this has finally taught us that we actually can have much more rapid ways of disseminating information and have that be a very viable way to actually get shit done. What was really cool is we saw, early after the sequence was published, as very early in the pandemic, that this was a beta coronavirus. It was very much related to other beta coronaviruses we had seen, like SARS-CoV-1 and MERS-CoV. In fact, when we also saw the structure, which came out just weeks later, 5th of February 2020, the structure for MERS-CoV was often very similar for different kinds of proteins in the virus, there's not many, to SARS-CoV-1, which we knew something about. In fact, the protease structure was almost identical. Stephen was telling me earlier this morning, and it was really remarkable how similar the active sites were in both sequence and in the three-dimensional position of their structures. So obviously a protease, especially one that's essential for the virus life cycle, got us thinking about maybe this could be a good target for oral antivirals. The protease, for example, is essential because of this part of the life cycle where the virus translates this polyprotein that has to be cleaved in 11 different places to liberate the different functional parts of the virus so it can go off and become infective. If it has to cleave in 11 different places, you might also imagine that it's very difficult for resistance to emerge as long as you're staying within the active site, because you have to modify both the active site and all of those 11 cleavage sites simultaneously in order to have the virus be viable. There's binding sites that you could occlude, so you can prevent it from doing this, and you could disrupt the virus life cycle by just having an orthosteric ligand that competes with substrate. Of course, we had no idea that Pfizer was working on making Paxlovid orally bioavailable at the time. It was a peptidomimetic. It was very hard to formulate. In fact, there had been a lot of these peptidomimetic small molecule inhibitors for this protease known, but the two challenges with this is, number one, these things are basically just slightly modified versions of the peptide substrates where they've put some different chemical groups on them. They're okay for intravenous injections, but certainly not something you can easily formulate as an oral pill. It took, in the end, Pfizer $1 billion and 350 people in order to do that in a very short amount of time. So we didn't have the time nor the money to do that. And the other thing is that these are covalent inhibitors. That means that they have the potential to bind any cysteines that are accessible. And you can definitely have covalent drugs, but you have to worry about off-target effects because you don't want them labeling other cysteines that they shouldn't be because that can cause off-target toxicity. So it takes some time to do that tuning, and we didn't think we had that time. So we decided that we would not focus on covalent inhibitors. We would try to make things that were much smaller, much more easily formulatable. The other problem was that there had been no approved coronavirus Mpro inhibitor drugs for humans. However, there had been success in cats. This is my cat that spent a lot of time next to me during the pandemic. These cat inhibitors, which were also intravenous at that time, were successful in actually curing certain coronavirus infections of cats. So we at least knew that these kinds of inhibitors would work in mammals. So being naive scientists who use computers a lot, we thought, of course, we can use our computational acumen to accelerate this whole process of traditional drug discovery. We don't have 10 years to do this. Let's just do this as quickly as we can by using how smart we are. But fortunately, we also had a secret weapon, and that secret weapon is protein crystallography. So this is the Diamond Light Source in UK. It's right outside the Oxford Harwell campus. And the MX beamline has called XChem has this really cool high throughput automated fragment screening technology. What happens is that they grow these very small crystals, but very reproducibly. And then they use an echo dispenser, which is an acoustic droplet ejection dispenser that uses a jet of water to shape an ultrasonic pulse that ejects a nanoliter droplet of a small molecule in DMSO. And this sits on the surface of the crystal and gets pulled into the active site if it likes to bind into the crystal. And it's maybe 10 or 20% of the active sites get bound because these things are very weak inhibitors. They look like small fragments of real drugs, but by having a huge amount of automation and collecting a really large number of these structures, you can merge all the data sets and find even 10 or 20% populations where that ligand is sitting, and of course, how it binds, and get a structurally resolved picture of what this active site might like to bind in terms of chemistries. And that's extremely useful for structure-based drug discovery. So once you do this, you can get this picture that you could then hopefully use to accelerate the overall drug discovery pipeline. I just want to emphasize how quickly this happened. So Haitao Yang's lab was responsible for solving that first coronavirus structure I showed you, but they had to shut down due to COVID. So they sent the sequence to Marty Walsh at Diamond Light Source in Oxford. They managed to synthesize the plasmid, actually clone it, get protein, purify it, get crystals, and then screen 1,500 of these co-crystal structures with fragment screens, all in just a few weeks. It was an enormous effort from a huge number of people. And then they got the data online as soon as they could. They spent all their time solving and getting these structures online. And this was like much faster than waiting for a PDB deposition, of course, right? It's just pre-printing. They have this wonderful portal called Fragalysis, where you're able to actually visualize this in your browser. You can download all the data. You can look at these things or different combinations of fragments. And he just tweeted about it. That was the most rapid information dissemination process available. What was cool about this is if you look at this data set, it really does map out this entire active site. So this protein is functional as a dimer, but if you just look at one of the monomers and zoom in on the active site, you could see that all of these fragments nicely fill all of the pockets that these side chains would fill when this peptide substrate would lay across it. Here's the cysteine that would be catalytic. The peptide goes here, and all these S's are different pockets that those side chains would fit in. And if we look at pairs of these compounds, you can see that a lot of the moieties do overlap in space very closely. So of course, structure-based or fragment-based drug design has been around for a long time, but the idea is that you could maybe synthesize these into a much more potent compound and jump ahead several steps from those early stages of high throughput screening or any of the other technologies you would need to use to modify or to find an initial potent lead that you can then optimize. Now, the problem was, even after fragment-based X-ray drug discovery being a mature field, we still don't have a consensus on what's the best way to actually put the data together to make compounds. And so near London, at Weizmann Institute of Science, who had been collaborating with all of us at the same time, said, what if we just tried everything at the same time? Now, this sounded crazy, but also maybe not too crazy given the circumstances. So we had to come up with a cool name, the COVID Moonshot. That sounds pretty cool. We had to come up with a principle of how do we get all of these people to collaborate? We can't spend time signing agreements about who owns the IP, so let's just make everything open. All of the science will be open, all the data will be open, and we're going to say there's just nobody's going to take a patent on this. We're just doing this for the benefit of humanity. So we've set up a website, and this came from, again, finding more folks on Twitter that were willing to help out. Alpha Lee at Cambridge, who had started a new company called Chodera, said, my company could put together this new website. We'll have people input their ideas. They can upload smile strings, or they can sketch them here if they're just sketching ideas. And then we're basically going to ask people, you design it, we'll figure out how to make it. And so Near tweeted this out, calling all med chemists. We released 60 structures of fragments bound to MPRO. We're crowdsourcing ideas of what to do next. And what happened next was absolutely staggering. So we thought a few folks would maybe use their fragment-based drug discovery tools and run these and submit some ideas, but this got very widely shared. There were lots of folks in industry who were sitting home alone looking for ways to contribute. There were lots of other academics who wanted to use their own tools. Medicinal chemists donated their time. We had a huge amount of response from a lot of really amazing people in terms of actually designing new compounds that we could advance. So we got more than 7,000 designs from over 350 different designers. It was a huge amount of stuff that we then had to wade through. Some of those designs were really thoughtful. People thought, these are fairly well elaborated, potentially soluble, potentially good protease inhibitors. Others maybe had other kinds of rationales. I apologize, I'm just going to read the design rationale that we captured at the bottom. They said, these compounds have similar Hansen solubility parameter values with other protease inhibitors. Well, that might be true. At least they're using a model to drive their decision-making. That's good. These were a little bit more, caused me to wonder. But the rationale says gold is thiophilic. These can be sourced from E molecules and tested versus MPRO, especially as Orofin acts on COVID-19 cells, and they cited a paper there. So at least there's, again, a rationale for why you might prefer gold-containing compounds, even though I don't think these will work. I don't want to get anywhere near these compounds. I fear they might be highly explosive. Someone says by eye, these are tiny molecules. That's true. These get really interesting. I'm looking for common, inexpensive, widely available compounds, preferably volatile, that humans already safely inhale and, if possible, enjoy inhaling. All right. These are just carbon, I'm not sure they'd be all that safe either. And then, my favorite one was this one. I used random numbers to find this compound, is what the rationale says. I use random numbers in my drug discovery work too, so I felt sympathy, but I don't think this is going to work out. And then, of course, the design rationale is common sense. And the other notes say, I'm sure it works on a dish at least. Well, it certainly helps us cope with the pandemic. That's for sure. So we had to find some way to sort through all of these design ideas. And we work on structure-based drug discovery techniques. So we used virtual screening methods based in OpenEye to very easily screen these things, and especially to look at whether or not they actually elaborated on the design fragments. So the nature of the game is to make sure that you have these inspiration fragments that you list to say that I'm supposed to make something that builds on this fragment. And so ideally, you're looking for things that redock with those moieties in the same position. You can filter out a bunch of bad ideas this way, but you also need a way to synthesize these compounds. So we're very fortunate that AI and ML tools and drug discovery have advanced to this point where things like molecular transformers, which rely on much the same technology for language translation for Google Translate, are able to help us retrosynthesize and find ways to synthesize these compounds using building blocks that are already in stock at CROs like Enamine, WuXi, and Sai. And we've been super lucky because folks like Enamine, which is a premier synthetic chemistry CRO based in Ukraine, have been a stable partner with us throughout this entire effort, doing things either for free or at cost. They've been phenomenal folks. So using this technology and their huge building block libraries and a huge amount of Enamine donated chemistry time, we were able to make 850 compounds in just a few weeks, which is an enormous amount of compounds. And it was the same kind of thing. We took all the data, we put it back online. You could browse all the different binding affinity curves we had for our biochemical assays. These are protease inhibition assays. We were also able to get structures for basically everything that was synthesized. And this is quite unique. Because of Diamond's high throughput beamline, we could basically soak everything back into those crystals, put all the structures up as well. So we could not only have the biochemical activities, but also the structural hypotheses for how these things actually bind. And it worked. That's the amazing thing. So we got so many compounds that were actually measurably potent. None of those fragments had any kind of measurable potency better than 100 micromolar, but a bunch of these design ideas synthesized these fragments and actually came up with really nice elaboratable scaffolds that we could do chemistry with that had measurable detectable activities, which is super amazing. This one was about 25 micromolar. But we had several different series of these. So we not only had this 3-aminopyridine series where we also had made several different variants of this. So we tried to do SAR in parallel. We also had these OOGIs, which are one-pot syntheses, which you can make 4 different substituents on at the same time. And then these the 2 different benzotriazoles on the right, and then the quinolones on the left over here were other backup series, some of which had been seen to have similar scaffolds had SARS-CoV-1 activity before as well. So that gave us more confidence that those might be useful. So we picked the 3-aminopyridine to work on, and maybe these others could be backups if we have money to work on them. Unfortunately, we don't have any money to work on any of these. And of course, we're not at the point where we can actually say that this is a drug, right? This is just a weak hit that we can start to elaborate, or maybe an early lead. And we've gone from here, where we're just finding which target to work on, only to here, where we now have the beginning of a lead optimization phase. Now, when you're doing lead optimization for drug discovery, you still need some goals, and you need a process by which you can actually prosecute these discovery programs. I've been very fortunate to go to Santa Fe, New Mexico in 2020, the last meeting of the year in March, and ran into Ed Griffin, who had just given a keynote about how computational chemists and folks who do ML or AI for drug discovery could actually work with real medicinal chemists in a productive way. So of course, we immediately roped him into our effort, and he became our lead medicinal chemist for this. He's an ex-AstraZeneca oncology vet who now runs a consultancy called MedChemica. And he set out this basic strategy for how we're going to work. We're going to aim for small molecules. We're aiming to make things that are orally formulatable. So we want to keep them tiny. We want to keep them simple. We're going to avoid these peptidomimetic-like compounds, even though they're potent. We aim for potency and then worry about covalency only if we need to, because then otherwise we'd be spending a lot of time working out these off-target issues. We're only going to go for SARS-CoV-2 now. We're not going to worry about other coronaviruses at the moment. We can come back to that later. So what you do in a real drug discovery program is you write down your objectives for what you're trying to accomplish. This is called the target candidate profile, or TCP, which says, okay, I need to hit the target biochemically. I also need to make sure it gets into cells and do the thing in cells. It needs to hit a decent enough spectrum of what I'm working with. If it's going to be orally formulated and orally delivered, it needs to be soluble enough. And also, when it's interacting with plasma proteins and other things and being metabolized, it has to stick around at high enough concentrations that it actually has antiviral effect over the EC9 coverage over the EC90 for that time in between dosing periods. And then you've got this entire set of toxicity or safety things you need to worry about. For example, if you inhibit hERG, then you're going to have a bad time because you're going to have a bunch of adverse cardiac events, and you don't want people dying of heart attacks when they're taking your drug. To actually meet those objectives, you need to set up some sort of assays that are surrogates for these real objectives that you're trying to meet. And all you're, in this meeting these objectives, what you're trying to do is to persuade someone that they're able to take this and spend millions of dollars to convince the FDA that it's time to do clinical trials on humans, that it's safe enough to do that. So you organize these things into tiers where all of the cheap, fast stuff is in this first tier where you're looking at biochemical activities. And then if it passes a certain criteria, you're going to do more expensive and more time-consuming assays. And of course, during the pandemic, access to BSL-3 facilities where you could actually do these antiviral assays was very limited. And so those were in our second tier. And then other stuff that involves a lot of rodent or animal work, then you want to put that into the third tier because you're sacrificing animal lives. It's much more expensive. It's much more time-consuming, the time it takes to get those measurements back. And so you run many cycles of this through these lead optimization cycles where you make some hypotheses, you design some molecules, you synthesize what you can, and then you actually do some of those fast experiments. If they pan out, then you promote them to the next stage, and you hope you get all the way to the point where you can actually ideally de-risk the biology, or even go all the way to making sure that this is something that you could develop into something you're willing to put into humans. So you run through these cycles many, many times, right? And this is where our lab designs these structure-based discovery technologies that help us predict what these potencies were. Surely we should be able to shove this in at this point to speed up lead optimization. So a lot of these cycles look like this. We have a hit molecule, that's the initial one I showed you. You find a retrosynthesis route where you can install different R groups at each stage, but you want to do this at the last stage, because if you make a lot of this one intermediate, then a good chemist at Enamine can make a dozen of these analogs per week. And you could do anything that fits with this palladium catalyzed coupling in this last step. So there's like 15,000 building blocks that they have in their library that you could use. The chemists are very conservative. They propose a very small amount of modifications that really feel out the structure activity relationships, or SAR, in this binding site, with the idea that they'll make progress one step at a time, but it takes them many cycles through. So our job is, what have they missed? Are there things here that we can make easily, that we can just add basically for free, that will let us hop to better potency right away? And so what you can do is you can dock in a bunch of all of these building block design ideas and make structural hypotheses. But there's 15,000 of things that we can make. We use this technology called alchemical free energy calculations. Folks like Dave Case and Darren York have also contributed to making these really practical for industry to use on practical timescales. What happens is that you turn one ligand into another ligand through a thermodynamic cycle where the free energy around this cycle has to add up to 0. But you use all of these clever statistical techniques to compute the things that aren't easy to do in real experiments, where you turn one ligand into another ligand by actually modifying its Hamiltonian. And there's a huge amount of statistical inference work that's gone into this. I don't have time to get into it, but it's a really cool field, and it helps set up your graduate students for going to work in machine learning and statistical machine learning areas where they can put a 1 or a 2 in front of their salary. You can use these technologies not only for driving potency, but also for driving things like selectivity or predicting the emergence of resistance. So will this mutation of a few atoms on the protein side change the affinity for the ligands? And that's a huge problem in cancer, like I've referred to. You can also use them for optimizing biologics, both in terms of optimizing binding affinities and in optimizing thermostability, so partitioning between a folded and unfolded state. And then there's a really cool set of new, because we're getting structures for things like HERGs or more SIPs, thanks to emergence of cryo-EM and other structural biology techniques, these are going to be useful for all sorts of ADMET properties as well in the future. We build a bunch of open source tools. A lot of it's based on OpenMM. Actually, I think we're up to 1.5 million downloads for OpenMM so far, just from CondaForge. This is a GPU-accelerated molecular simulation and modeling software that's just easy to install via Conda. It's free, MIT licensed. Anyone can use it or build on top of it. There's a bunch of industry programs where you don't even know you're using it under the hood. We build all of these middleware layers on top to do really cool algorithms, and then different applications that are specialized to different domains, like whether you're trying to get things that permeate bacterial pores or trying to optimize small molecules. We're also lucky that we're able to work together with these consortia, like the Open Force Field Consortium, where we have over a dozen different pharma companies, as well as some federal funding, working together to build open source tools that pretty competitively help us in different areas that are difficult for drug discovery. So Schrodinger may, for example, have encrypted force fields where you can't even see what's going on under the hood and you have to pay for access. But here, everything is open. You can even rebuild these force fields yourself if you want to. You can take your own internal data sets, add them to the public data sets that we've generated, retune the force fields, and over many generations, as we produce these public force fields, they actually do get better. So we're now on several more generations since in making these much more accurate. This model was so successful that we decided to replicate it. We built this organization called the Open Molecular Software Foundation, which is set up with the same general counsel as the Linux Software Foundation. So it's a good, healthy open source initiative that can act as a fiscal sponsor for different organizations like Open Free Energy, which is trying to commoditize a lot of these free energy tools, as well as things like OpenFold, headed from Mohammed Al-Qarashi's lab, where companies can then build versions of AlphaFold based upon their internal data, as well as actually going way beyond this to modeling protein ligand interactions. So when we did a little bit of work retrospectively, some graduate students and postdocs helped out. Dominic Rufa was instrumental to this, as well as Hannah, who's now at Charm Therapeutics, and Will Glass, who's now at Exscientia. They were able to show that the tools that we had at hand could actually retrospectively describe what's going on. There's lots of questions about protonation states and other modeling choices, of course, but it seemed to be good enough to try to use prospectively. The problem was we don't have 15,000 GPUs, right? We can't possibly grind through all of these using our internal resources. How do we do that? Well, we happen to be part of this Folding at Home consortium. It used to be, so it started in the year 2000. It was something like distributed.net or SETI at Home, where people would run a screensaver at home, where they would, when they're not doing work, would run this thing in the background and use their computing time to do actual science. And that science for us was running molecular simulations with things like OpenMM. And so we had this consortium of folks who were different laboratories around the world who were helping us engage with all these volunteers who are now typically gamers, who, when they're not playing computer games, they're turning their GPUs on, often because they need to heat their house. We had 100 petaflops of computing power, so we thought this is a huge amount to actually dedicate to this at the time. So we had a rotation student, Ariana, work with a couple of other graduate students to write a little blog post, and then somehow this got picked up by the news media and all sorts of other outlets. And we had over 1 million volunteers join in just a few months. So soon we were looking at 1.5 exaflops of computing power, which is more than some of the top 10 supercomputers. And if you would cost this out on AWS, it would cost $6.8 billion a year. We did not have 100 times more people, so we were just often trying to keep the servers from exploding, but we did our best in trying to use these resources for many different things. Some of it involved collaborating with folks like Vir, who had sotrovimab approved as one of the antibody therapeutics for a while. We were helping them try to understand this interface between the SARS-CoV-2 RBD with ACE2, the protein that is part of your cells, that's the receptor for these things. We had now a huge global collaboration where not only did we have volunteers around the world contributing their computer time, but we also had all of these other laboratories that were donating their time. We still don't have any funding for this project, so we have to get folks donating synthesis time. We have to have folks donating assays as well. So we had also never run these calculations on folding at home, which was one difficulty. Normally, when you run these calculations, you use these Hamiltonian exchange methods. Any of you who do molecular simulation, it's a very clever method for ensuring you get adequate mixing amongst all the different alchemical states. It's a big problem of sampling from posterior distributions. It's a very clever method, but very hard to do if you don't have any kind of coupling or parallelization between things. There had been this other non-equilibrium method that had been used. Where this came from was this guy, I sat next to him at Berkeley, Gavin Crookes. During his PhD, he invented the Crookes fluctuation theorem, which is one of the most impressive laws of nature. It replaces the second law of thermodynamics with an equality that relates systems driven out of equilibrium to exact equilibrium free energy differences. And it turns out they had used this in single molecule experiments when I was hanging around with the Bustamante laboratory doing these laser trap experiments, unfolding proteins one molecule at a time. It was super cool to see, but we knew we could use the same theory to pull these ligands. So you start in equilibrium and then you pull them from one state ligand A to another state ligand B. So you basically turn on this part in a non-equilibrium manner. Then you park it there for a while till it reaches equilibrium, and then you go back again. And from doing many of these cycles in parallel, you could reconstruct the overall free energy difference of binding. So now this is used quite regularly in a lot of things, like including OpenEye, where I'm on their SAB. You can do this in parallel in a few hours for drug discovery. We put up a cool web page. We have a progress bar. That's really important. People feel like they're chipping in if their progress bar goes up, thanks to their help. We did a lot of simulation time, like 10 milliseconds per week, which is a lot for those of you who are used to doing simulations. We still had no funding, so we had a GoFundMe, and people could contribute money to pay our chemists eventually. And it seemed to work. So in retrospectively looking at this, we made a number of different modifications that we would not have made without the free energy calculations. The overall correlation with the experiment was surprisingly good. We were using as much of the experimental data as we could at the time to help leverage to make better predictions, but some really cool insights emerged from this as well. So if you look at the entire library, these are better compounds, these are worse compounds in the current design. Most of the library is dumb. I mean, this is a fully, like, it's literally anything we can make with the building blocks. So most of it is not helpful in many of the cases. There's only a small fraction of the compounds that are predicted to be better. We also had humans putting in design ideas in parallel. So this is Ed and Ralph. Ed and Ralph produced many fewer designs, but they're clearly shifted towards more potent designs. On the other hand, there's more in the overall library. So even if it's not as good, there's still quantitatively more better design ideas that come from looking at the entire library, which I thought was fascinating. We put all of the data online into these web pages so that you can see in this real-time dashboard what's floating to the top, how much of the computation has been done. And you can look at these metrics of whether or not the calculation you really believed. We look at all these transformations and which was predicted. Sometimes design ideas from the human chemists would float to the top. So this is a very adventurous transformation that cyclizes this, so locks the stereochemistry in with the spiro, and then by doing so, you're able to reduce the conformational entropy for binding. And it turns out that the prediction was very good for this, and it turns out that actually worked, and even the structures that we predicted and uploaded to fragalysis seem to very well match the binding mode that came through crystallography in many of these cases. So in the end, we actually achieved our goals. We went all the way to the point where we could produce something, the first molecule that met our target candidate profile goals. We kept going, of course, to try to find better compounds to put into the preclinical program. But all of this is up on our preprint. Here are the main milestones, where essentially, after all of that work, you kind of make an elaboration here to improve potency, then another elaboration here, and then you keep going to improve some other aspects. So it ended up being quite serial in that respect. But we profiled it extensively, put it all up online. There's over 180 contributors. This is an amazing paper because it has 2,220 compounds made and tested with all of the experimental data disclosed, everything you might want for a drug discovery program. over 850 X-ray structures just for this one target, right? Last I checked, this was something like 30% of all SARS-CoV-1 structures in the PDB at the moment. And then 400 potent compounds you get from this. So people can literally buy them from enemy. You don't need material transfer agreement. You don't need to synthesize them yourselves. You can go to the catalog number that's in the paper, and you can just order it from enemy and to do your own experiments. The really interesting thing was that because we were trying to put the data online, we were managed to submit. So it takes a while to submit these things to the PDB, but some of the early structures did get in very rapidly thanks to the work here. One of those early structures was cited in figure 2b of the discovery of ensitrelvir, a new SARS-CoV-2 MPRO inhibitor that is approved in Japan. This was done by Shinogi, one of the premier Japanese pharmaceutical discovery companies. They used this to generate a pharmacophore model that they then used to fish out compounds in their late-stage pain programs, and had compounds that they knew that very well that had exceptional PK. So instead of 6 pills a day and 8 for 5 days for Paxlovid, because you need ritonavir boosting, which then means you have 8.5 pages of drug-drug interactions, which excludes a huge amount of the vulnerable population from being able to take Paxlovid. This is a once-a-day molecule with much, much fewer liabilities. It's a phenomenal molecule now on fast track for FDA. And it's just absolutely exceptional and really cool to see that they were able to use open data rapidly disseminated through the PDB in order to discover this compound. Just super cool. And so even if we're trying to accelerate antiviral discovery in the open, it does actually accelerate commercial discovery as well. The really cool thing too is that all the liabilities are going to be very different. We're eventually going to see emergence to resistance to Paxlovid. We haven't seen a lot of it yet, but the liabilities for nirmatrelvir versus ensitrelvir versus our compounds are very different because they contact different parts of this binding site. We were then able to finally get some funding for this project after we had finished it, to get to the point where now we need to take this into humans, right? We need to get it to the point where ideally we can find a generics manufacturer and go direct to generics with this therapeutic. There's a path to that. We ended up getting some funding from Wellcome Trust and the Access to COVID Tools Accelerator from the World Health Organization to get it to the point where we can do first in humans, and then we need the partner after that. Things are moving very rapidly, or not as rapidly after the discovery part. So it took about 18 months to get to the molecule candidate. Things get very slow because there's not a lot you can compress in the preclinical phase, but there is still this path straight to generics. And given that I just read that Paxlovid might go to $2,500 a course next year, having an ultra low cost available to anyone and globally and equitably accessible and affordable therapeutic could still be very useful. When we were kind of taking stock of what we learned in this, we wrote this little perspective or comment for Nature. Zooming in on this, the compounds that we made here are equipotent against SARS-CoV-1 main viral protease, right? Pfizer stopped their program in 2004 because they couldn't figure out, the population for SARS-1 patients had gone away, but there was no incentive to take it to phase 2 readiness, where we would have been a lot faster, or all the way to therapeutic. We could have done this in 2004 for a couple $1 million, right? To get all the way to the point where we had an orally available small molecule. It could take a few more millions of dollars to get to the point where it was phase 2 ready, had pills manufactured, had a clinical trial protocol. That may have extinguished the epidemic before it became a pandemic, right? So, when you're thinking about that, plus the fact that we're going to have this a lot more, right? You might be noticing that there's a lot more pandemics now. It turns out that global warming is forcing animal reservoirs into closer proximity to humans, or new proximity to humans that haven't happened before. So they called this the pandemicine, because we're just going to see this a lot more with global climate change. So if you think about how you prevent a future pandemic, like what do you really want to do? Well, it's very similar to what's the best way to win a race, right? You want to run fast, but you also want to start near the finish line, right? And we could do that. We actually have the opportunity to do that. We can not only develop it, these are really easy drug discovery programs. Coming up with an orthosteric inhibitor for a protease, very, very easy. Especially because the proteins look so different from human proteins, unlike cancer. So you can develop a technology platform that moves very rapidly. You can also start close to the finish line. So we could do all the work up front. We can get as far as we can by exercising this platform so that we have all of these great broadly neutralizing or broadly antiviral inhibitors to start with. And if we do need to tailor them to make second generation inhibitors, we have the technology to do that as well. Fortunately, this gentleman convinced the president in 2021 the beginning of his administration to set aside $3.2 billion for the antiviral program for pandemics to fund a few different activities, including these antiviral drug discovery centers. And I'm super lucky that we were able to actually apply and become one of these antiviral drug discovery centers. We got the first 3 years of funding for the 5-year program. We were able to spin up what essentially amounts to a distributed biotech for antiviral discovery. We have several different classes of targets we're working on, both coronaviruses, which are responsible for SARS, MERS, and COVID, flaviviruses, which are things like dengue and Zika. There's not any global market for this, right? But these are coming to the United States. They're already in Florida, for example, dengue cases. And then picornaviruses, because polio is back, baby, right? We've got these enteroviruses and polio resurgence. We need to worry about antivirals for these because we don't have any. So we do target identification in a way that's also, you have to very carefully engineer small molecules that are not going to be immediately susceptible to resistance. That's a huge problem with HIV, for example. We have target enablement, which is using this diamond light source at scale to really generate huge numbers of structures that enable these targets with fragment screening, often with multiple different variants from different viruses. We then can accelerate a lot of the downstream elaboration. Instead of doing this by crowdsourcing, we can use AI and different computational chemistry algorithms to do that. There's also some cool covalent targeting. Then we can do accelerated lead optimization using our planetary scale computing. And then finally, we have funds to actually take these all the way to clinical readiness and then hopefully translate them as far as they can. Like for dengue, there's actually 100 million people a year that are infected each year in the world. So we can actually move that forward to approval if we can. And unlike a normal drug discovery company, where you look and see what their pipeline is, what they're working on, and have just big blobs about what stage they're in, here we're able to share all of the data openly. You can drill down into every stage of this and see all of the data we've generated up to this point in time. You can see everything we're working from in terms of plasmids. You can go get them from AdGene. You can find all of the protocols we're using for assays. You can look at our assay cascade, see exactly what criteria we're using. You can find all the molecules. You can buy them from Enamine to do in your own experiments with. You could grab all our structures. All the data is available. What's really cool about this is that this is a huge opportunity for folks that are developing new methodologies. We're sending millions of dollars through this cycle every year, so we're able to put different methodologies into place and see how well they do prospectively, which is hugely challenging in our field. So, one of the things that I'm excited about, this is an amazing team, but it's just a bunch of humans making decisions over Zoom calls, right? This is not the econometrically optimal way to make drug discovery decisions. Instead, we can leverage this amazing growth in structural data, especially when we're looking at how to make these things broadly neutralizing across different viral species. Over the last 10 years alone, there's been over 100,000 new structures deposited in the PDB. It's amazing how fast this is growing. We can leverage all of that, including with AlphaFold-like technologies, to speed up this whole process. We use these experiments as surrogates for the real thing. So we can also use computational experiments as surrogates. This looks like amortized inference, where you're doing the cheap stuff first and the more expensive stuff later, but you're trying to build models that later predict what goes on in the more complicated things. So there's a variety of different ways we can build these models with different machine learning classes, structure-based methods or non-structure-based or mechanistic-based methods. And then just like when you're playing chess on a computer or Go, you can think several steps ahead. We hardly do this in drug discovery right now. We do this implicitly by saying, well, I really like this compound, so I'm going to make more of it now so I can put it into rat PK if I need to. But it never uses a model underlying that. That's what I'd like to change. And so we can think about all the technologies needed to really move the human away from being in this loop of choosing exactly what molecules get synthesized to thinking about what are my objectives? What are my strategies that I'm going to use to achieve those objectives? Why are these models working or not working? And really think about automating this process, this platform that I alluded to go as quickly as we can in case we need it during an emergent pandemic. It'll also make regular drug discovery cheaper too, which is great. We can use funds to actually build antivirals to do for a good cause as part of this process. So with that, I want to thank, this is just a fraction of the folks who have contributed. I don't even have the final list here. You'll have to go to the manuscript on bioRxiv to see all of the folks that have contributed to the work that I've talked about today. I especially want to thank all of the chemists in Enamine who have been with us since the start. These are phenomenal people who are incredibly resilient and extremely hardworking. And all of my laboratory too, and all the funders that have let me focus on this for the last couple of years, because it's been immense fun, and I hope to be able to continue to contribute over the next few years. So with that, here's all of the data, and I'll thank you for your attention and happy to take any questions.
Thank you, John. That was it was a tour de force, and it shows how important open data are. And it also shows you how important the PDB is. And I want to emphasize that every one of those structures that was determined at Diamond is almost certainly now in the PDB. And I thank the people who worked on, particularly Zukang, who worked on our high throughput data collection mechanism to bring the data into the PDB. So questions, comments?
And thank you for doing that, by the way.
Yeah. Thoughts? Dennis?
So yes, great talk. Thank you very much. And I love the idea of crowdsourcing for help. And it's just amazing. I think the pandemic really highlighted that people are always willing to help when there's a dire need for it. I think I missed it, but when you got all these crowdsourced ligands, how did you filter out all those bad ones automatically?
That's a great question.
I'm sure, and I'm also curious what the percentage was of the ones that were valuable or potential of value versus those that were not possible at all.
So it was a combination of two things, right? One of them was, is it a terrible idea? And for us, the computational approach to that was to ask, if we redock this compound, especially using these shape-based overlays or substructure-based overlays, you can do all of this in OpenEye Toolkit very easily. Does it recapitulate the binding mode, and does it clash with can it physically fit? And does it establish this or throw in this hypothesis that they articulate, because we captured which design ideas they started from, which design fragments. The other thing is it synthetically feasible, right? So it also has to be very rapidly feasible. So ideally 1 or 2 step, not something that's going to take 5 steps and somebody has to optimize the route. So the intersection of those two is what led to our initial bolus of what we can make.
That was really cool.
Thanks.
I have a question about resistance and the emergence of resistance. And while your original intuition that you would need to make 11 changes, so there's greater selective pressure to keep it, to prevent resistance from emerging is right. I think we've learned that the protease doesn't need to be as active as the wild type, right? And so you can take a hit and just be 10% active, and that's still enough, because you really have nothing against you and once you get in. So given that, what is the current status of resistance and what sort of general themes are emerging? Can you comment on that?
So first of all, it's terrifying if you look at the Cove Glue database or any of these databases that aggregate all the mutants you see, because you'll see things like the catalytic protease being mutated away or catalytic cysteine being mutated away. And you're like, oh my goodness. So a lot of those are probably sequencing errors or just non-viable virus that people have captured. There are a bunch of mutations that people have seen, and Rami Amaro had this great paper showing that some of them are actually already resistant to nirmatrelvir, which is the active antiviral of Paxlovid. So that's worrying, but we haven't seen a lot of spread of those resistance mutations. People expected much earlier we would see this spread, but it hasn't quite happened in the clinic yet, even though you can see these, especially if you do viral passaging, you see the emergence of those mutants as well. That we have to be careful about that, because we don't want virus that's been mutated and is resistant to drugs to escape laboratories. So now the NIH is preventing that from happening. It's interesting. I can't explain why we don't see the amount of clinical resistance that we expect, but what we've been following right now is there's 3 different things. Well, number one is the substrate envelope hypothesis of Celia Schiffer. She's done these wonderful visualizations to say, are you staying within where the peptides sit, right? The next thing is it that there are certain kinds of cleavage events that are intramolecular, where if it doesn't happen, if you're able to inhibit it, then it can't get rescued by trans cleavage, right? So what's worrisome is these RNA can mutate and then directly make protein. So if you have one copy of mutated genome, it can make protein that can rescue the rest of the protein in the cell. That's for a trans cleavage. That's very bad. So Carla Kierkegaard has helped us find which of these cleavage events are cis cleavage, and this protease does have some cis cleavage required events that help us if you inhibit any of these things, it'll make toxic products that kill the cell essentially. So that helps us as well. And the third thing is that we're investing a huge amount in deep mutational scanning. I don't know if you've seen any of Jesse Bloom's work for the spike protein, but these deep mutational scanning methodologies identify like the fitness of various different mutations that have been tremendously useful in identifying when we're going to see escape and which residues, exactly which residues and transitions we should avoid. So we're using all of that data right now to try to design inhibitors that are not going to elicit resistance. That's a great question.
Brenda and then Greg.
Great talk, thank you. So my question is, looking at what worked retrospectively based on the lessons learned, were you able to go back and identify prediction, the algorithm, the workflow, the calculations, or places that can be improved?
That's a great question. So many people submitted with so many different design approaches, right? That we've tried to do some retrospective analysis, and I think the latest version of the preprint has some of this in part of a figure, but there's just so much data there. Like it's really difficult to go through manually these. It was over 800 different submitters, or 300 different submitters, and over thousands of molecules. That work still remains to be done. So I'm very curious if anyone wants to dive in. All the data is available. You can download it. All of the rationales are in this table. It would be really cool to see, which methods did the best at really fusing these together. We don't have an answer for that yet. There's so much science that's left to be done from this open data package. But that's a really great question that I'd love to know the answer to. It was not a well-controlled experiment, I will say, though. Like what we'd really like to do is to have really well-controlled experiments that allow us to make sure that we're not, using any more data than we think we're using. This is why, these blind predictive challenges like T3R, sample, and now cache have been so instrumental, and KELP as well, which uses data from the PDB to continuously integrate. This is what I'd love to see is if all these computational methods were containerized and then subjected to KELP-like continuous evaluation challenges for data that's flowing through the PDB or through Kemble, this would be an amazing, unbiased, well-controlled way to actually assess the different utility of different methodologies and actually making good predictions. So I think that's the kind of well-controlled experiments we need, or we need a way to put them in process in our real drug discovery programs to see how well they do against each other. It would help with the platform, yes, yeah, absolutely. And if we can containerize the methods too, then you can easily bring them in and deploy them in antiviral discovery or deploy them to industry. My dream is that, if we did have like a common container component repository, you give a DOI in your paper for your containerized tool, it has a standard API, we could pull it in and use it internally and then advertise, here's how well it did on this antiviral program, right? It would make everything a lot easier. Yeah.
Thank you for the very interesting talk on a very important subject. You were mentioning that the humans do better percentage-wise in terms of designing hits, but the computer, because it has such a high number, high throughput, you get an absolute larger number. Of those good hits that the computer can predict, going to the next step, how many of those you just mentioned easily synthesizable? How many fit those category, and how many fit the category of having proper PK characteristics?
Yep. Okay.
Which and do you think do the algorithms need to be tweaked to sort of improve on that end of it also? Can you improve that?
You've zeroed in on the exact weakness here. So that, first of all, these are all synthetically feasible because they're part of the library. I mean, like they're everything that's compatible with that last synthetic step. So pretty much everything in that library has equal chances of success for synthesis, and is all very high. The problem, as you've articulated, is that we're just prioritizing for affinity here and not for any of the other objectives that you need in your target candidate profile. And that's what needs to change, right? We need to have good surrogate models that we can pop in and continue to retrain with new data that hit all of those objectives simultaneously, so you can prioritize the selection of molecules, both to reduce your uncertainty in the models, as well as to make better compounds. So you want to have this trade-off between exploration and exploitation, like in Bayesian bandits.
So there are a couple of questions in the chat, and I'm going to ask you to respond to those.
Happy to, yeah.
When you're done, if you don't mind.
Great.
I just wanted to take the chairman's prerogative and just make a few observations, if I may. Could we go back to your 3 inhibitors slide for a second? So yes, this is the one. So John alluded to this. Despite the fact that nirmatrelvir was first to market. It does have significant liabilities because it has to be co-administered with ritonavir because it's very rapidly degraded in the liver. And ritonavir blocks that, but that means that certain people who take statins, for example, take anticoagulants, can't take nirmatrelvir. So there is a significant need for more than one drug. And it is, the fact that Shionogi is presumably going to have something to market very soon is a very good thing. Otherwise, you're stuck with molnupiravir from Merck, which is a repurposed hepatitis C virus protein, which isn't that effective, but it doesn't have the same liability. So these are important considerations. The second point I just want to contradict something you said about access to nirmatrelvir in the third world. Though in those economies, they don't respect US patents, so they just make the drugs and sell them generic, so it doesn't matter, right?
Has that happened? I know.
I mean, in China, only the very rich buy the branded molecule. Everybody else buys the knockoff generics. And there may be issues with that, but that is the reality for a quarter, somewhere between a quarter to a fifth of the world's population. And the third thing I want to do is to relentlessly plug my own talk coming on November the 8th, which will actually go through and detail the nirmatrelvir origin story, which actually begins with work we did at SGX Pharmaceuticals in the early 2000s. Put the data into the PDB immediately, and that enabled Pfizer to move forward in the early 2000s with the SARS-CoV main protease inhibitor. The problem there was a failure of the free market, as John alluded to. The consortium that you put together represents, I think, an important lesson for people as to how failures of the free market can be circumvented. The free market capitalism is a great thing when it comes to making a better mousetrap. It's not a great thing when it comes to making an emergency pharmaceutical and budgeting funds for something that might happen. And if it does happen, it'll be terrible, but no one knows the actual probability. No one knows the size of might. What's the probability that something like that will happen? Well, after SARS-1, no one knew. Arguably, after MERS, everybody who was an expert knew there's going to be another coronavirus and it could be bad. And the third time we were extremely unlucky. So I'm going to talk about this more on the 8th. Let's thank John for an amazing talk and the contributions that he and his team have made.